is Gabriella Vavala from the Cedars Sinai Pacific Palisades in California in the US. And the title of her talk is Interactive Effects of Prenatal Drug Exposure and Socioeconomic Status on Early Brain Connectivity. Can we please have her slides up?
Hello. So today I will be presenting our research examining the interactive effects of prenatal drug exposure and socioeconomic status on functional connectivity at birth. So for some background, resting state fMRI studies in infants and children with prenatal drug exposure, or PDE, show altered functional connectivity in limbic regions involved in reward processing and emotional regulation. And additionally, SES has been linked to altered functional connectivity development in the first year of life. In particular, lower maternal education is associated with poor neurodevelopment. However, little is known about the combined effects of PDE and SES on functional connectivity at birth, so we used resting state fMRI to examine the main and interactive effects of PDE and SES on functional connectivity at birth. To do that, we leveraged two data sets, and data set 1 was collected from 2008 to 2014. It included 52 drug-free controls and 63 infants with prenatal drug exposure. Dataset 2 was collected from 2016 to 2020. It included 28 drug-free controls and 81 infants with prenatal drug exposure. The infants were exposed to similar substances, and this graph shows the distribution of substances within each dataset. Dataset 1 was enriched for cocaine exposure, and dataset 2 was enriched for opioid exposure, which is why it's a bit higher here. And the infants in dataset 2 had greater overall drug exposure than the infants in dataset 1. We used maternal education as an indicator for socioeconomic status, and we used education instead of income because income was thought to be less representative of true SES for our sample. This is because a lot of the mothers were in residential treatment facilities and therefore were not receiving income at the point of data collection. And looking at the distribution of education, the substance-using mothers had lower educational attainment than the control mothers, and the mothers in dataset 2 had greater educational attainment than the mothers in dataset 1. We think this is likely due to the time gap in points of data collection. Resting state fMRI scans were collected at 2 weeks of age. And first, we calculated functional connectivity. To do this, we derived functional connectivity measures using the Neonate Functional Parcellation-Based Atlas by Shi et al. 2018. And this atlas has 223 ROIs. For each ROI, we calculated mean reference connectivity across 100 drug-free neonates from the UNC Early Brain Development Study. And then we calculated deviation scores for each participant in datasets 1 and 2. And we did this because some drugs increase connectivity, whereas other drugs decrease connectivity. So assessing magnitude instead of direction prevents potential cancellation effects. Next, we ran a whole brain heat map analysis examining the main effect of PDE, the main effect of maternal education, and the interactive effect of PDE and maternal education. We ran linear regression, functional parcellation-based linear regression, controlling for maternal depression and birth weight, and we generated heat maps showing the percent of significant effects within each ROI's functional connectivity map, thresholded at p less than 0.05. We generated 3 heat maps, one for the main effect of PDE, one for the main effect of maternal education, and one for the interactive effect of PDE and maternal education. And since the two datasets had different levels of maternal education and drug exposure, we wanted to see if the significant effects were shared between the 2 heat maps for datasets 1 and 2. To determine that, we calculated overlap heat maps, and for each ROI, we identified the significant connections shared between the 2 datasets and divided by the total number of significant connections across the 2 datasets. Lastly, we ran an ROI level analysis, and using the heat maps from step 2, we identified top-ranking ROIs with the highest percent of significant effects. Then for those ROIs, we identified which connections were affected by PDE, maternal education, and the interaction. Looking at the heat maps, we first examined the main effect of PDE on functional connectivity in dataset 1, and we observed widespread network disruptions in the frontal, temporal, and subcortical regions, including the caudate. And these regions show up as red on the heat map, meaning that approximately 9% of the connections in these ROIs were affected by PDE. Next, we looked at the main effect of maternal education, and we observed effects in the frontal, right temporal, and right occipital area. When we looked at the interaction, we observed widespread network effects in the frontal, temporal, and left sensory motor area. Switching over to dataset 2, we observed the main effect of PDE in the right frontal, left temporal, and mid-cingulate area. Again, looking at maternal education, we observed some overlapping effects in the right frontal, left temporal, left mid-cingulate, and then distinct effects in the left sensory motor area. And lastly, looking at the interaction, we observed some effects in the right temporal, right frontal, and right mid-cingulate area. So next we looked at the overlap between the pairs of heat maps across the two data sets. For PDE, we observed overlapping effects in the temporal, left mid-cingulate, and occipital areas. For maternal education, we observed overlapping effects in the right temporal, left anterior cingulate, and thalamus. And lastly, looking at the interaction, we observed overlapping effects in the right frontal area. As mentioned, we ran an ROI level analysis, and we found that PDE had the greatest effect on the right superior temporal gyrus. When we looked at PDE's effects on this ROI's connectivity, we observed a decrease between the right superior temporal gyrus and the left superior frontal gyrus compared to controls. And we found the greatest effect of maternal education in dataset 1 on left Heschl's gyrus. Collapsing across the two groups, higher maternal education was associated with decreased connectivity between left Heschl's gyrus and the right inferior temporal gyrus. We found the greatest percent of effects of the interaction on the left inferior temporal gyrus. For the control group, higher maternal education was associated with decreased connectivity between this region and the right superior temporal pole, whereas for the PDE group, higher maternal education was associated with increased connectivity for these regions. For dataset 2, we found that PDE had the greatest effect on the left fusiform gyrus, and we observed decreased connectivity between this region and the right superior temporal gyrus compared to controls. We found that maternal education had the greatest effect on the left superior orbital frontal, and collapsing across the two groups, higher maternal education was associated with decreased connectivity between this region and the left middle frontal gyrus. Lastly, looking at the interaction, we observed the greatest effects on the right inferior temporal gyrus. For the control group, higher maternal education was associated with decreased connectivity between this region and the right superior temporal gyrus, right superior temporal pole. And then looking at the PDE group, we found that higher maternal education was associated with increased connectivity for these regions. And we found widespread effects of PDE maternal education and the interaction on neonate brain connectivity. These effects were primarily in the frontal and temporal areas, and approximately 10% of these effects in the frontal and temporal areas were reproducible between data sets. In conclusion, maternal education can alter the effect of PDE on neonate functional connectivity, and we believe that future studies examining the effect of PDE on functional connectivity should include environmental factors like SES and education. And I want to take a moment to thank everyone in the GAL lab, in particular Dr. Janelle Liu, who I worked very closely with on this project, and it would not have been possible without her, and Dr. Wei Gao for his guidance. And then I would also like to thank our collaborators and sources of funding. Thank you very much for listening.
Thanks so much. There's no questions on the app, so if there's no burning question from the floor, we might move on in the interest of time, because we did lose some time. Yeah, okay, well, thanks again, Gabriella. So the next